So in this EdPuzzle, we're going to talk about what happens when you have sort of larger scale problems with chromosomes. I'm not talking about single point mutations, we're just changing one base pair, but actually when you have chromosomes break or get transposed, so they swap places with other pieces of a chromosome, or in some cases where they don't separate properly during meiosis and you end up with extra copies of genes. So the first type of chromosome error we're going to talk about are errors that occur during meiosis. So these can cause large-scale chromosome problems, meaning your broken chromosomes when they're pulled apart. During anaphase, it is somewhat violent. You're yanking the copies of the DNA apart. So in some cases, they get broken, or you have them not separate properly, and so you end up with daughter cells that have extra copies and daughter cells that are missing pieces of information. And this can lead to a variety of different kinds of developmental disorders. And in lots of cases, birth defects or that are so severe that they may lead to, a miscarriage. Basically, the body says, hey, there's something wrong here, and this is not going to go forward. So non-disjunction refers to the process where either homologous pairs or sister chromatids not separating normally during either meiosis 1 anaphase or meiosis 2 anaphase. So depending on when it happens, whether it's meiosis 1 or meiosis 2, you're going to have a gamete that receives 2 copies of the same chromosome, and of course one gamete that has neither. In general, missing information is worse than having extra information. That's just sort of across the board. So missing a chromosome, of course, then you're missing that set of instructions. So you don't have any instructions on how to build any of those proteins or express those characters. And so that's something that's generally a fatal condition. So here we have a situation where we can see non-disjunction has occurred here on the left during meiosis 1. And so we had one homologous pair separate correctly here at the top, right? So then we have one of each, right, into each daughter cell and there. And then, of course, you had the second homologous pair not separate properly, and both pairs went to this daughter cell. And so then you have the other daughter cell is missing that information. So for this one, it's a fatal condition here for this cell with the missing chromosomes. The other one that got an extra copy, that may or may not be a fatal condition depending on what those genes code for. Here on the right, we've also had another situation where meiosis 1 went okay. Everybody separated the way they were supposed to. But then you had non-disjunction occur in meiosis 2, and therefore you have, again, one pair that gets one each, so haploid gametes like they're supposed to be, and then non-disjunction occur in this one, and so now this one has an extra copy. So assuming that it's not a fatal outcome, if you end up with an individual that has an odd number of chromosomes, and it's typically for just one particular chromosome. We're not talking about whole extra copies. We have another term for that. But aneuploidy refers to an individual who has one extra chromosome or is missing a particular chromosome. And again, this results from fertilization of gametes in which non-disjunction has occurred in one of the parents, right? So we have a normal set of gametes that are haploid merging with another set of gametes which are in which one of those chromosomes you either have an extra copy or you're missing a copy. So if a monosomy or if you're a monosomic zygote, then you are an individual that has only one copy of a particular chromosome. In many, many cases, this is a fatal condition because, as I mentioned earlier, missing information tends to be deadly rather than having extra information. The only monosomies that are really viable in humans are ones in which you're missing a sex chromosome. If you're missing any of the other chromosomes, then that's a fatal condition. So if you're missing any of the autosomes, then that typically is a fatal condition. And then, of course, a trisomy, therefore, would also be you have an odd number of chromosomes, right? You're supposed to be diploid. You're supposed to have 2 copies. And in this case, you have a 3rd copy of a particular chromosome. And there are several trisomies that are not fatal. You have just an extra copy of a chromosome, and those individuals are just a little bit different. But they have some challenges, but they often function just fine, having that extra information. For example, one common trisomy is Down syndrome. So these individuals are diploid, of course, but have an extra copy of chromosome 21. So we can see that here on this karyotype here at the bottom. Right, here's a chromosome 20, and then here's 21. You have 3 copies. So again, extra information is not as big of an issue as missing information. So these individuals sometimes have developmental delays, learning disabilities, but for the most part can hold down a job and function fairly well in society in general. They're very valuable members. They just happen to have an extra chromosome. So the frequency of this occurring in children increases with the age of the mother. This is a correlation that no one's been able to concretely explain in terms of exactly what happens in her cells. I think other than just increasing chance of mistakes being made as you get older, and just kind of the machinery starts to wear. I don't want to say once it starts to wear out, but as you get older, you're increasing chances of mistakes being made in general is generally true. Okay, so as I mentioned earlier, non-disjunction certainly is not unique to autosomes. It certainly can happen in sex chromosomes. And these are, again, are more likely to be non-fatal or have very limited side effects, mostly because, the Y chromosome carries very few genes on it outside of the SRY gene that codes for testes production. The X chromosome does have a significant number of genes on it, and so if you have any issues with that one, you're more likely to have some side effects of that. So in terms of non-disjunction, generally we have what's called Klinefelter syndrome. This is an extra X, so in individuals their genotype would be XXY, so they have 2 X's. And a Y, so they have the Y chromosome that makes them at least genetically male. They tend to again have very normal development. There are some physical characteristics. If you know someone has Klinefelter, you go, oh, there's those characters. But honestly, you couldn't pick anybody out of the population and say, oh, that person has Klinefelter syndrome. In fact, most people with Klinefelter go through their lives not knowing. They may have, so for example, a learning disability, but again, there's a lot of people who have learning disabilities that don't have Klinefelter syndrome, so there doesn't seem to be any kind of correlation there. They just usually find out when they have trouble having kids because, of course, they have an odd number of chromosomes. And so that means that maybe half of their gametes are not going to be functional. And so they usually go to a specialist, reproductive specialist, and they say, oh, by the way, did you do you know that you have an extra chromosome, and that's why you're having trouble conceiving as fathering a child. So so that would be again a trisomy with sex chromosomes. The only monosomy that's viable in humans is Turner syndrome. So again, all you're missing is a sex chromosome, And so individuals, of course, are female. They have an X, and then they're missing the other one. So obviously they're sterile, right? They don't have another chromosome to pair with during meiosis. So that's a no-go. Phenotypically, though, they're female, and again, completely normal in their development, other than the fact that they, can't conceive children. They have an X, though, so they have all of the genes that are on the X chromosome. They have at least one copy, so again, that information is present. So really, if you're going to have a monosomy, it's missing information. X is probably the safest one to be missing, because of course in females anyway, female mammals anyway, one of the X's is deactivated in all of our cells anyway, right? It's only one X gets expressed at any given point anyway. So if we only have one X, well, then all the genes that are on that get expressed, and problem solved. Okay, so polyploidy is different from aneuploidy. Aneuploidy, we're dealing with a situation where non-disjunction has occurred during meiosis, and you're ending up with an odd number of chromosomes for one particular chromosome, right? So again, chromosome 21 or chromosome 3 or something like that. You have an extra, or you're missing one. Again, missing tends to be fatal. Polyploidy, on the other hand, is a situation where you have an entire extra set of chromosomes. It may be a third extra set, a fourth extra set, a fifth extra set, something like that. So typically that's when you have, you've had non-disjunction happen, but on a bigger scale than just one chromosome. It's a whole set that hasn't separated properly, or you've had an error in DNA replication, and you've copied extra copies during that process as well. So there's one way that you can get there as well. Polyploidy is extremely rare in animals. I think I can think of a frog species that's polyploid, but really not very many other examples. Having an entire extra set basically tends to change what species you are. And so again, we'll talk about this more in 51 when we talk about speciation mechanisms. Again, animals having an extra set is usually a no-go. Plants, on the other hand, can self-fertilize. And so they actually tolerate polyploidy very well. So you can have non-disjunction occur in a plant as they're making their gametes, and then fertilization, they self-fertilize. And so that's another way you can end up with an entire extra set. You have a non-disjunction occurring, you have an extra set of chromosomes, and then you merge that with another gamete that also has an extra set of chromosomes, and you can end up with several extra sets. Again, that's something that in animals and other eukaryotes is generally something that's fatal. For some reason, plants tolerate this, having extra sets of chromosomes very, very well. In fact, most of our domestic plant species are food species that we use. Wheat in particular, I can think of strawberries, for example, are mostly octoploid, so they have 8 copies, extra copies. So they, clearly tolerate that just fine. That extra information does not affect their function at all, except, of course, to make them a separate species that can now no longer interbreed with their parent species. In fact, it's thought that up to 80% of our current living plant species are formed or were formed as a result of polyploidy occurring. So that's an extra set. So polyploids, again, that extra information is not as bad as missing information. Polyploids tend to be more normal in appearance than aneuploids. You can't really tell if an individual is polyploid unless you look at their genetics. Having an entire extra set, you kind of balanced. You have extra information all around rather than just one extra set. Okay, and finally, I mentioned we were talking about errors in chromosomes with regard to non-disjunction during meiosis, but also other kinds of errors such as broken chromosomes. So what happens when you have your chromosomes lining up right on your metaphase plate, or during the process of crossing over in meiosis, that you may have crossing over occur, and then you're pulling those chromosomes apart, and that can be somewhat violent. We also have errors that have occurred during DNA replication that were not caught by our spell check function that we're going to look at later on when we talk about DNA replication. So it's possible, for example, to have a section of the DNA copied multiple times. So this is gene duplication. This can be a great source of variation, actually. So again, having extra code, depending on which genes are being duplicated, may or may not actually be a problem. It really does depend, as we're going to look at later on when we talk about cancer. Having extra copies of, for example, the genes that are making growth factors and are encouraging cells to keep dividing and dividing and dividing, having extra copies of those genes is generally a bad thing in terms of pushing cells down the road to cancer, whereas duplication of other kinds of genes may not be a problem at all. Translocation involves moving a segment from one chromosome to another. So for your genes for eye colored end up on a different chromosome, and they switch places with another basically non-homologous pair or non-sister chromatid, which you don't want to have that happen, right? You don't want chromosomes that carry genes that don't code for the same traits to be swapping places. So this is sort of like crossing over, only it's not supposed to happen. And therefore, you've got really garbled instructions. And quite often, again, this is if it's not fatal, it can lead to some pretty severe birth defects or cancers in some cases. Inversion is kind of interesting. It's something that does not happen very often at all. And this is essentially where you have a section of DNA that gets flipped end over end. So again, possibly an error during crossing over. And now your code is effectively backwards. And that's also, again, something that is generally going to be a source of significant problems in terms of developmental issues, because those, of course, that code then can't be read properly. And so you can't make the gene products. So depending on which sections are inverted, you could end up with some pretty major problems. And then, of course, lastly, if you just cut a section out. So this again may be a result of I'm pulling my chromosomes apart during meiosis, and I kind of yank them apart and I break off a section, and it just sort of gets dropped and never is not reattached. So that again, that section of the chromosome is now missing. And so again, that information is missing, which is usually pretty bad. Depending on which genes you're talking about, but it can be quite detrimental to have that missing information. So just for example here of a translocation situation where again you have genes being swapped between non-homologous pairs of chromosomes. Here we have a switch, I think, between chromosome 22 and chromosome 34. Part of 22 ends up on chromosome 34. So it's not deleted, but it's been moved to a different chromosome. And so again, it's likely not going to be read correctly, and you have a problem with a broken chromosome, and then the end that gets stuck on to another one. In this particular case, it contributes to chronic leukemia. And of course, it's chronic because what you have is a genetic error. So it's not something that's easily fixable. It's something that's going to be a recurring problem. And of course, finally, in this course, we are generally focusing on nuclear DNA. When we talk about our genome, we're mostly referring to the genes that are contained on our chromosomes that are found in our nucleus. But we, of course, have to acknowledge that part of our genome is actually genetic material that's found in our plastids. We have a mitochondria, right? If we were a plant, we would have both mitochondria and chloroplasts, and each one of those actually have their own circular genome. It's a circular DNA molecule that is very similar to the genetic material in bacteria. So again, we think that these are ancient bacteria that started living inside our cells and inside the cells of plants, and so therefore they carry their own genes with them. Some of those genes have actually migrated into our nucleus, so we actually have mitochondrial DNA as part of our linear DNA. But what we don't have, of course, are the instructions on how to make more mitochondria. So if we lose our mitochondria, our cells have no ability to make any more, and they die fairly quickly without that massive, ability to convert glucose to ATP using cellular respiration, right? So we refer to this as our extranuclear genes. So these again are the genetic material that's in our mitochondria. And of course, the genes that we have in our mitochondria, all of our mitochondria that we have in our cells comes from our mother. And part of that is, again, she's stacked the egg with everything that baby fertilized zygote is going to need to jumpstart cell division once fertilization happens. But part of it, people would say, well, there's mitochondria in the sperm cell too. Why don't we have a mix of both? And it's essentially because the mitochondria in the egg are basically very jealous and territorial, and they seek out the vessel. They have vesicles that seek out the mitochondria that are coming in from the sperm cells and essentially digest them. They secrete digestive enzymes and basically break down the paternal mitochondria right after fertilization. So the paternal mitochondria get wiped out by the maternal mitochondria. It's like, wow, that's pretty dirty pool there. Pretty vicious. And so again, all that's left are the maternal mitochondria. So of course, this is how folks can do, for example, if you don't have, and then we'll talk about this later on when we get to forensics, but when you have a DNA sample that you don't have any nuclear DNA, so for example, the shaft of our hair, there's no nucleus to that, but you do have mitochondria. So if you can extract that DNA from the mitochondria, then you can determine whether someone is the daughter of, so-and-so. You can identify their maternal lineage. You can't identify it to an individual, but you can say, hey, this is the offspring of so-and-so because they have the same mitochondrial DNA. So it is a way of doing some form of genetic testing, but again, it's not individually identifiable. Okay, so that's the end of this EdPuzzle. It's not so long, and I didn't figure you would mind.